Lymphocyte development and survival are determined by signals received through their antigen receptors. Equally amazing as the generation of millions of specificities of lymphocyte antigen receptors is the shaping of this repertoire during lymphocyte development and the homeostatic maintenance of such an extensive repertoire in the periphery. How are the most useful receptor specificities selected? And how are the numbers of peripheral lymphocytes and the percentages of B cells and T cells kept relatively constant? The answer seems to be that lymphocyte maturation and survival are regulated by signals received through their antigen receptors. Strong signals received through the antigen receptor by an immature lymphocyte cause it to die or undergo further receptor rearrangement, and in this way, self-reactive receptor specificities are deleted from the repertoire. However, a complete absence of signals from the antigen receptor can also lead to cell death. It seems that in order to survive, lymphocytes must periodically receive certain signals from their environment via their antigen receptors. In this way, the body can ensure that each receptor is functional and regulate the number and type of lymphocytes in the population at any given time. These survival signals appear to be delivered by other cells in the lymphoid organs and must derive, at least in part, from the body's own molecules, the self-antigens, as altering the self-environment alters the lifespan of lymphocytes in that environment. Developing B cells in the bone marrow interact with stromal cells, while their final maturation and continued recirculation appears to depend on survival signals received from the B cell follicles of peripheral lymphoid tissue. T lymphocytes receive survival signals from self-molecules on specialized epithelial cells in the thymus during development, and from the same molecules expressed by dendritic cells in the lymphoid tissues in the periphery. The self-ligands that interact with the T cell receptor to deliver these signals are partially defined, being composed of known cell surface molecules complexed with undefined peptides from other self-proteins in the cell. These same cell surface molecules function to present foreign intracellular antigens to T cells. Lymphocytes that fail to receive survival signals and those that are clonally deleted because they are self-reactive undergo a form of cell suicide called apoptosis or programmed cell death. Apoptosis, derived from a Greek word meaning the falling of leaves from the trees, occurs in all tissues at a relatively constant rate in each tissue and is a means of regulating the number of cells in the body. It is responsible, for example, for the death and shedding of skin cells, the turnover of liver cells, and the death of the oldest intestinal epithelial cells that are constantly replaced by new cells. Thus, it should come as no surprise that immune system cells are regulated through the same mechanism. Each day, the bone marrow produces many millions of new neutrophils, monocytes and red blood cells, and lymphocytes, and this production must be balanced by an equal loss of these cells. Regulated loss of all of these blood cells occurs by apoptosis, and the dying cells are finally phagocytized by specialized macrophages in the liver and spleen. Lymphocytes are a special case because the loss of an individual naive lymphocyte means the loss of a receptor specificity from the repertoire, while each newly matured cell that survives will contribute a different specificity. And that's lymphocyte development and survival are determined by signals received through their antigen receptors. Have a great day.